Welcome to lab number 18, where we are still on the brain, but we are switching things up today. We are switching from that brain model to a sheep brain. So we're looking at an actual sheep brain right here. Lovely little sheep brain. They're kind of small. You can easily fit one in your hand. They're really not that big because, sheep are really not that smart. But we do see some good surface area in this brain. We see a lot of gyri. We see a lot of sulci. Of course, remember, gyri are the ridges. Sulci are the grooves. So we see them, plenty of those. Now we can also see just the shininess of this brain. You can really see it, especially in the sulci, you can see that shininess. And that shininess, of course, is due to the pia mater, one of the three meninges that wrap and protect that brain. The pia mater is your innermost meninx, so it is the deepest of the three. So we're going to go through this sheep brain. We're going to look at stuff on the superior view here. Then we're going to flip it over, look at some inferior view stuff. Then we're going to slice it mid-sagittally, look at some stuff on the inside. And that should just about do it. So let's start naming some things. We do see the frontal lobe right here. That's the frontal lobe of the sheep, where it does all of its thinking, where it has its sense of humor, its personality. I don't know how much of a sense of humor or a personality a sheep would have. But there's the frontal lobe. We'll go back a little bit. We are in the parietal lobe here. Sense of touch, body awareness, of course, is one of the big jobs of the parietal lobe. And if we jump back to the back of the cerebrum, the back of this right cerebral hemisphere here, we got the occipital lobe, which of course is very much involved in vision. Now we also see that longitudinal fissure running right down there. That is going to separate the left cerebral hemisphere from the right cerebral hemisphere. Okay, what else do we have in this top view? Well, we can see the cerebellum. In fact, this whole thing right here is the cerebellum. There it is, very involved in coordination, in motor control. And this is the vermis. This whole big chunk right here is the vermis. The sheep have a relatively bigger vermis than us human beings do. So that's the vermis right there. Very nice. And we see the right cerebellar hemisphere here. We can see the folia on it, just like we saw in the human brain model. Of course, the left cerebellar hemisphere is over on the opposite side. All right. What the heck happened here? Well, I should have prefaced this a little bit. What we're about to do is we're about to take one hand. We're going to put one hand right here. So you're going to put one hand right here. There you go. There's one of your hands right there. It's your left hand, I guess. And there's your watch. Bum. All right, so you got your left hand right there. We're going to put your right hand more or less right here on the cerebellum. And a little bit of the spinal cord, touch of the brainstem. There we go. So you're going to put your hands like that, and then we are going to pull the cerebellum down so we can open up this transverse fissure right here. We're going to open up this transverse fissure so we can take a peek inside. So that is what we are going to do. Now I should advance to the next slide, and lo and behold, we've done it. So just to orient you, this is the cerebellum right here. Here's the vermis. Here's a hemisphere on the right, hemisphere on the left. Here we've got our cerebrum right here. We're basically in the occipital lobe of the cerebrum on each side. And we've opened up the transverse fissure, right, separating the cerebrum and the cerebellum. And we've done that. We can see some midbrain right here. Now, before we go on to the next slide, let's do some counting real quick. 1, 2, 3, 4. So we count 4 things here. All right, if we got 4 things, do you have a guess as to what we are actually looking at? Did you say it? Did you know? It is the corpora quadrigemina, the 4 twin bodies on the superior midbrain in the sheep. Okay, so you got that corpora quadrigemina, which, if you recall, has a pair of superior caliculi on the top. Very much involved in visual reflexes, visual coordination, like keeping your eye on something and moving your head and your body to follow it. So that is this right superior colliculus. Of course, the left one is over here. It has an L on it now. So you got our 2 superior caliculi right there. Can you guess what we're going to do next? I am hopeful that you can. Yes, there it is. That is an inferior colliculus right there. There's the other one over there. Inferior caliculi are very much involved in auditory reflexes, like my favorite one, the startle reflex. Hopefully you have tested that out on someone to make sure their inferior caliculi are working. All right. Excellent. And hey, we flipped it over. We're looking at the ventral view of the sheep brain. The ventral view. Now, in this ventral view, there are a lot of things we can see. We can see the brain stem. Basically right here. There's the brain stem. We can see the spinal cord right here. We got some cerebrum on the side here. Some cerebrum on the side. Some temporal lobe. And we can see a little sneak peek of the cerebellum right there. So let us go over some of these things as well as some of these whitish structures that we see. Some of these many, many structures that we see. So let's do some of those. All right, we're starting with the left olfactory bulb. So right there, that little white region right there is the left olfactory bulb right there. That is where the olfactory nerves come in from the nasal cavity, bringing that sense of smell towards your brain. Now, not only do we see an olfactory bulb, but we see the next thing on the olfactory pathway. And I switch sides here for some reason. We see the olfactory tract right here. There's the olfactory tract, taking that sensory information towards that medial temporal lobe so the olfactory cortex can start to interpret those smells. All right, so that really takes care of olfactory stuff. Let's now do some optic stuff. Now, in this particular sheep brain, like this is a gorgeous sheep brain, but we don't actually see the optic nerves. The optic nerves would be coming out right here, coming out right here, but we don't see them. They've been cut away. What we do see is the middle of our X right here. So realize if the optic nerves were still here, we'd have an X, right? We would have an X if the optic nerves were still here, but we don't have the optic nerves. It doesn't mean we don't have the middle of the X, and the middle of the X is the optic chiasm. That's where some of your optic signals switch sides, so we can have a sort of contralateral situation with vision. Okay, after the optic chiasm, what do we got? We got the optic tract. So there's the right optic tract right there. Here's the left optic tract over here. Optic tracts are taking that visual information to the thalamus where it can be filtered, sorted, and sent on its way. Okay, we're moving our way posteriorly here. Let's keep on going. Just posterior to the optic chiasm. So right here, there's a little tiny opening. It's hard to see. Little tiny opening right there where the infundibulum would attach. Recall that the infundibulum is what connects the hypothalamus to the pituitary gland. We don't see the infundibulum or the pituitary gland here, but we're going to know where they would attach. All right. If we move posteriorly again, we got this roundish guy right here. This is the sheep's single solitary mammillary body. So optic chiasm, infundibula, mammillary body, which is very much involved in smell, also involved in memories for you and me. Okay, and of course we have two. Sheep have one. Let's jump back a little more. We're on the midbrain here. Beautiful midbrain. Look at this midbrain right here on each side. Lovely midbrain. Fantastic midbrain, in fact. What else can we say? Well, on this midbrain, this is actually one of the oculomotor nerves right there. It would be nice if we could like lift it up and flap it around, but since it's a picture, we can't. But right there, that is an oculomotor nerve. Of course, the oculomotor nerves are cranial nerves number 3. They're going to be strictly motor. They're going to control almost all your eyeball muscles, as well as being involved in things like constriction and dilation of your pupil. All right. If we jump back, we got the pons. And one thing we noted in the brain model was the bulginess of the pons. And we can still see that bulginess here as well. The pons is really going to stick out. Pons, of course, literally means bridge, forms a connection between higher and lower nervous system structures. If we keep on going, we can see a cut trigeminal nerve. This guy right here is a cut trigeminal nerve. Remember, the trigeminal nerve is cranial nerve number 5. Cranial nerve number 5. It is a mixed nerve, so it has sensory and motor functions. It does both. It gets sensation from your face, and its motor job is to control the muscles of chewing. I know what you're wondering. Can we learn the names of those chewing muscles? And the answer is yes, absolutely, just not yet. Hold your horses on that one. We will get to it in good time. Okay, we zip on back. We got the medulla oblongata, which of course controls a lot of homeostatic functions, very much involved in maintaining proper heart rate, blood pressure, respiratory rate, etc. All right, guys, that might be it for this inferior view. Let's see. Yes, it is. So now we have a mid-sagittal section. We got a mid-sagittal section. We can still see the cerebrum right here. We got the right cerebral hemisphere here. We got some cerebellum back here. We got some diencephalon right here. And we got a good amount of brain stem. And we got some spinal cord down here as well. All right, we are starting here by noticing the corpus callosum. This whole thing here is the corpus callosum, the white matter that connects the right and left cerebral hemispheres. Now, if we find that corpus callosum, we can very easily find this flimsy looking membrane right here. That is the septum pellucidum, which separates the lateral ventricle in the right hemisphere from the lateral ventricle in the left hemisphere. All right. We have another bit of white matter that we can see. It's this guy right here. This is the fornix, which is going to connect your diencephalon to your cerebrum. We saw that in the brain model as well. Now down here, we see the optic chiasm, cut in section. I always like to start with the optic chiasm before I do the diencephalon. So here is the optic chiasm. If I find that optic chiasm, then I can immediately take a little diagonal hop. So optic chiasm is here. Put an X on the chiasm because chiasm means like an X. Then I hop up, and right here I have the hypothalamus, which we know is like the master control center of your endocrine system, your autonomic nervous system. It is involved in all sorts of homeostatic things as well. So we jump from the optic chiasm, whoop, right up into the hypothalamus. Then from the hypothalamus, we are going to jump again. So we got the optic, whoops, I went too fast. There we go. We got the optic chiasm right here. We got the hypothalamus right there. Jump from one to the other, then jump up, and we have this circular structure here. It is the interthalamic adhesion, relatively bigger than that interthalamic adhesion in my brain and yours. You might not even have one. I might not even have one. Not everybody has an interthalamic adhesion. Whoa. Anyhow, you know we're not all the same on the inside, just like we're not all the same on the outside, right? There is variability. Oftentimes it can be quite extensive. All right, there is the interthalamic adhesion, which is connecting the two halves of the thalamus. And of course, the thalamus is our sensory filter. So we're not bombarding our conscious self with every little thing that's going on in and out of our body. Now, once we find the interthalamic adhesion, we've basically found the thalamus. You can't see much more of the thalamus than the adhesion in this view. If I drop down, so I go straight down, I go from the center of that adhesion, straight down, boom, I got that mammillary body. Which, if you recall, when we looked at the ventral view, we went from optic chiasm to infundibulum hole to mammillary body. In this view, I like to drop from that interthalamic adhesion straight on down. All right, let's keep on moving. So if we continue our diagonal path here, we went from optic chiasm to hypothalamus to interthalamic adhesion. If we keep going up on our diagonal line here, that is the pineal gland right there, which of course makes melatonin, which is very much involved in sleep-wake cycles, as well as seasonal things like breeding cycles in animals like sheep. Now, once we find that pineal gland, we can then find its neighbor, and its neighbor will be in the midbrain. We'll get to that neighbor in a second. This is the midbrain right here. So if you think about the interthalamic adhesion as being the diencephalon, then you jump backwards, you're in the midbrain. And there's that neighbor to the pineal gland I wanted to mention. Really pretty big right there. It is the superior colliculus right there. So you find that pineal gland, hop back, and you got that superior colliculus. Now, once we find the superior colliculus, what do you think we're going to find next? It's the inferior colliculus, which is right there. Remember, the four colliculi make up the corpora quadrigemina, which are part of the tectum, which is part of the midbrain. And speaking of midbrain, we can see the passageway running through the midbrain right here. That is the cerebral aqueduct right there, connecting the third ventricle, which will be around the adhesion here, to the fourth ventricle, which is down here between the cerebellum and the brainstem. So that is the cerebral aqueduct. Keep on going. We got the pons. Check out the bulginess of the pons right there. So if you find that midbrain, if you find an aqueduct and you keep going, you got the pons. And now we're jumping back up to the cerebellum, and we see its beautiful white matter here, which we know is called the arbor vitae. White matter of the cerebellum. And then we have that fourth ventricle that I mentioned earlier, between the cerebellum and the brainstem, connected to the third ventricle up here by the cerebral aqueduct. And the fourth ventricle will continue downward into the spinal cord, the central canal of the spinal cord. There we go. As well as the fourth ventricle has exits for CSF to get out into the space around the brain and cord, and that space is called the subarachnoid space. All right, there's our medulla oblongata looking very nice right there. And guys, that was a fast one. I probably went too fast, but kind of do that sometimes. My apologies if I was going too fast. You can always pause and rewind and watch things again. So we will see some sheep brain pictures on the third lab exam, so be expecting those, okay? All right. I will see you guys next time. Bye-bye now.